Okay. I think I'll go ahead and get started with Dr. Smith's introduction. That will take a couple minutes, and by that time, any late participants should be with us. So we're happy to have Dr. Stephen Smith with us today to present a seminar. And Dr. Smith grew up on a farm in the UK and got his PhD from the University of Birmingham. In 1977, he came to the US to work with Major Goodman and Charles Stuber, after which he began a 35-year career with DuPont Pioneer. Dr. Smith has received the Henry A. Wallace Award for Revolution in Agriculture and DuPont's highest scientific recognition. And Stephen can correct me if I say this wrong, the Lavoisier Medal for Scientific Achievement. He's a fellow of the Crop Science Society of America and currently is an affiliate faculty member at Iowa State. And in his spare time, he plays the organ and serves on committees with the Des Moines Symphony Orchestra and the board for Living History Farms. So that's my introduction of Dr. Smith. And I think, Dr. Smith, I will let you introduce your title and your seminar. So thank you for being with us and speaking to us today.
Well, thank you for that nice introduction, Jody. Thank you, Jody and Jian Ming, for having me talk to you all this afternoon, and thank you all for being here. And when Jody asked for the correct pronunciation of a French person and asked an English person how to pronounce French, it's almost or more dangerous than asking an American person to pronounce Lavoisier. I think you were as close as I can get, so that may not be good enough for the French, but it's good enough for me. All right, so we're going to look at estimating, and I'm saying estimating, and you'll understand why as we go on, the global usage of Corn Belt germplasm. And there's lots of implications, and that's going to end us up with a lot more questions, I hope, and discussion at the end than I've got answers for, because I think these questions need to be asked. Okay, now, all right, now it doesn't want to work. All right, so here's the content of the talk. Many of you, most of you, perhaps all of you have never heard about or even remembered the 1970 southern leaf corn blight. We're going to briefly discuss that. We're briefly going to get us all on the same page by talking about the origin and global distribution of maize, background of Corn Belt dents. We'll have a quick look at which countries are the leading producers in the world, and we'll then look at how different ways in which you can characterize maize genetic diversity and the challenges in actually doing that for diversity on farms in cultivation. And then we'll get through to these questions, discussion topics, which are the two big bullets towards the bottom here. Now, in 1970, most all of corn germplasm was in T cytoplasm because producers were using cytoplasmic male sterility. And the T cytoplasm was associated with the vulnerability of the whole plant to the fungus that caused southern leaf corn blight. And the losses in yields were huge back in 70 and 71. You can see that Illinois and Iowa were both producing at that time they occupied 18% of the US corn area each. And the yield losses from southern leaf corn blight were between 26 and 31%. So this is serious stuff. The overall economic loss was $7 billion in today's terms, or you could look at it another way. It was 20% of the total crop value. And if that was looking at the 2020 harvest, that would be worth $12 billion. So this caused a lot of concerns and led to an investigation by the National Academy of Sciences, and they produced this book called the genetic vulnerability of major crops. It didn't deal just with corn, it dealt with the major US crops. And this was published in 1972. And so 2022 is the 50th anniversary of this document. And briefly, the document said, for all the major crops, genetic diversity seems to be decreasing through the way pedigree breeding is being done. And that was one of the conclusions. And they argued for a change. And one of the reasons you have the genetic enhancement of maize project is because of the stimulus to exploring new genetic diversity that was spurred by the corn blight. Okay. Now, briefly, looking at maize, I'm sure you all know that maize was domesticated from teosinte in Oaxaca, Mexico, about 8,000 to 10,000 years ago, and spread north and south in the hands of explorers, farmers, travelers, and led to several different forms from selection in those particular environments. And basically, in the Americas, there's some 300 different races have been described, but only 6, about 2% of those are economically important to producing maize. And those are the Mexican dents, the corn belt dents, which are the corn that is grown in the United States, basically, and elsewhere. Tucson's, Caribbean flints, northern flints and flowers, and Catitos or Argentine flints. And amongst these, it's corn belt dent, which is by far the most economically important genetic diversity resource of maize. Now, after Columbus bumped into the Americas in 1493, this was after the Vikings apparently actually got to this continent by quite some way. Corn then went global, and here briefly summarized are the routes that corn has taken going around the world. It got to Europe very rapidly, 2 or 3 years after Columbus bumped into the Americas. And it actually got to China quite early too. And there are several different routes into Europe, Africa, and Asia. And there's been there's different ways of getting there, and there's different ways, different times in which materials have got there, right through till the 20th century. And here briefly is current global cultivation of maize. Orange red is the most densely cultivated areas of maize. And you can see obviously the Midwest, Mexico, and China. But corn in Europe is big too, and in many parts of Africa and Brazil and Argentina. So this is telling you nothing you didn't already know. Here is a chart of the top 20 producer countries, at least the average of 5 years from 2015 to 2019. And you can see by far the 2 largest global producers are the United States and China. 27% of global production from the US, 19% from China, Brazil 6%, Argentina 3%, and then it starts falling, and you very rapidly get down to 1% and down. And the top 20 countries collectively are producing about 72% of global maize. Now, let's look at the corn belt dents, because this is the most economically important germplasm base of maize, and it's what is effectively constitutes the United States germplasm base. And as we will see in this seminar, not just the United States. Now, there are two origins. So hybrid corn has a sort of hybrid ancestry, which is quite interesting. It's mostly the southern dents genetically. These came into the United States relatively recently in the 16th century into southeast part of the North American continent through the hands of Spanish explorers. Their genetic background actually could do with some research, I think. It looks to be pretty diverse. There's lots of potential ancestry in there. Given this is the most important component of the most important economic race of corn, I think there's some projects here that need doing to see how the southern dents actually compare to the rest of corn. The other parentage of the Corn Belt dents are the northern flints. Now, the northern flints arrived, corn actually arrived into North America around about 3,500 to 4,000 years ago. And the northern flints went right up to New England, and they were cultivated there around about 1250 AD, a bit earlier than that, but not really as a very major crop to begin with. So those are the two major components: the southern dents, mostly a fair bit of northern flint, and more recently there has been a little bit of what we would call exotic germplasm introduced into US corn, not very much. So Maize imago, for example, is one of the more widely known exotic pieces of corn that came in from Argentina. And it's sort of now a sort of natural part of Corn Belt dent. So when I talk about Corn Belt dent, I'm going to talk about the southern dents mixed with the northern flints and a little bit of other germplasm that's come in that's now a usual part of Corn Belt dents. So let's have a quick look at maize production increase in some of these countries from 61 through till 2014, 2018. And production has increased fourfold in the US. It's gone up 12-fold in China. And it's gone up 11-fold in Ukraine. And that was just between 1992 and 2018. Now, of course, that doesn't tell you why it increased. So to give you some, or how it increased. So to give you some more ideas, here's the area harvested, changes in the maize area harvested over that same period. So the USA production went up fourfold. Its area of in which it's planted went up 1.5-fold. So it looks like yield had something to do with it, the total production increase. China went up 12-fold. The area under production only went up 3-fold. So again, we're looking at yield as a major factor. Okay. And in fact, here are maize yield changes during that same period. So the USA maize yields went up nearly 3-fold. France went up 3-fold. Argentina 4-fold. China over 4-fold. Brazil 4-fold. South Africa 4-fold. Mexico nearly 4-fold. So there's big increases in maize yields. But now, of course, the question is, okay, so what's contributing to these maize yields? We tried messing around with these data to see if there was any evidence of a change in the trends during this time period. And it looked possible that there might have been some increase in maize yields from this linear in Ukraine, South Africa, Brazil, and possibly Argentina. There was little evidence for anything other than a linear increase throughout this period for the USA, China, and Mexico, and France appears to be plateauing. And that might be because, I mean, these are complex reasons as to why these changes are happening, but that may be because of weather, and it may be because of government regulations on use of pesticides and fertilizers and such like. That's a complicated, it's complicated to understand why these rates change. That isn't the main topic we're talking about here, but just gives you some background. Now, how can we characterize genetic diversity? Well, there are several ways. Looking at the United States, there are pedigree data, but there are some problems here because the inbred parents of hybrids, which of course are what's grown in farmers' fields, are maintained as confidential information. So it isn't publicly known for most hybrids what the parents are. The pedigrees of inbred lines are known if they are patented or PVP'd. There's two problems there, though. One is the ability to track those pedigrees to publicly known founders has really declined in the past 20 years, such that the public information on those pedigrees, it's only covering about half of the total pedigree. So that is a bit of an unknown. And as mentioned in the first bullet, the relative usage of inbred lines as parents of hybrids is confidential and not disclosed publicly. So when an inbred line is patented, usually its pedigree is revealed, but only perhaps half of it that you can understand in terms of relating it to public germplasm. And then, of course, you've no idea which hybrids or how many hybrids or what acreage that inbred line was used in. So pedigree data are getting increasingly not useful. Now, marker technology is pretty useful, but bear in mind that markers are just surrogates for measuring diversity. They're mostly anonymous. The ones that characterize diversity are mostly anonymous with respect to known gene function. And so, okay, we can see trends using, I don't know, several hundred to several thousand or tens of thousands of markers. These days, it's SNPs that are the preferred method. But in terms of relating that diversity to actual gene function, that's a different issue, as you all will know. When we go back to 1970, 1972, when that corn blight came out and that report came out, it's important for the students to know there were no genetic markers available at that time for corn. Genetic markers only started to become available in the middle to late 1970s with isozymes, and up until about in the late 1980s, David Grant will give me the exact date because he was developing RFLPs at Pioneer at the time. We actually went from 20 markers to 150 odd, and that was a great breakthrough. And so you can see that it's only fairly recently with SNPs when we're talking about economically getting a lot of genetic marker data for tens of thousands of locations on the genome that marker technology has really come into its own for many things, but for characterizing diversity, although we don't understand the diversity. Now there are some, so you might think, well, this is great. All we have to do is go out and fingerprint the hybrids that are grown in the United States on a regular basis. And by golly, we build a database and we can track diversity and everything's going to be wonderful. We can monitor what's going on. Life is not that simple. Okay, we're talking about SNPs now, but as I've said, it took a while to get to SNPs, and you went through isozymes, then restriction fragment length polymorphisms, then amplified fragment length polymorphisms, then you might have dabbled with rapids if you were unlucky, and then you went on to simple sequence repeats, and then you got to SNPs. So every time you have to build a new database. So that was a bit of a problem. So there was no consensus until we get to SNPs, where you can establish, okay, we're going to use these markers. Whatever the platform is, it won't matter. We're just using these markers and we can monitor diversity. So you might think, okay, we're in great shape now. Well, no, it isn't that simple again. The last survey of diversity of US widely used hybrids was published in 1992. I mean, this is 30 years ago, using RFLPs. You might ask, why on earth has no one published a survey looking at the diversity of corn using SNPs? I mean, it would be an obvious, great project to do. Well, if you look at bag tag licenses on the hybrids, you will find language that one way or another prohibits marker profiling for commercial and effectively for any purpose. So therein lies the rub. And then it's a bit more confusing because you have all these different brand names with different GMO stacks, but they may be based on the same or very similar genetic foundations. So that complicates that sampling issue as well. Okay, when a patented or PVP'd inbred line comes off its protection period after 20 years or before, if it comes off early, you can then obtain the inbred lines and you can do with them what you want if they're in the public domain, so long as there's not encumbered by any other patents on regulated genes, or for example. So, okay, all right, well, it's possible then to, and people have do this, to molecularly profile inbred lines when they come off PVP. And that's fine. The trouble is, of course, we get back to this issue that we don't know which inbreds made up which hybrids, and we don't know which hybrids were grown on how many acres, and therefore we can't translate that diversity meaningfully in terms of how much it was actually used on farms. So there's real big problems if you want to monitor genetic diversity in cultivation. Even if you want to monitor it 20 years late, you've got problems because you don't know which inbred lines were used in which hybrids and which hybrids were used on a lot of acreage. So this is a big problem if you want to monitor diversity. Now, so here looking at pedigree information. So this is looking at pedigree information for a set of inbred lines, and the company names are listed along here. We've got some time frames: 1980 to 84, 85 to 89, 90 to 95. And you can see we've got some colors here that reflect the sort of background those inbred lines were coming from a structure analysis of the pedigree data. So again, you can see that different breeders have different germplasms that they are using. So for example, in the 1980s, Holden's had, according to these inbred lines that were PVP'd, about equal use of B14, B73. They were not using much B73. They used a lot of Ohio 43. Pioneer in contrast, was using quite a lot of B37 and a lot of iodent Lancaster. And then you can go across the different time frames, and you can, at your leisure, you can see changes from other companies. Generally speaking, as time goes on, iodent gets more and more important. But here's the rub. These are just inbred lines that were PVP'd in that period. Again, we don't know which, if any, hybrids they were used in, how widely those hybrids were used. Perhaps inbred lines were also used that were not PVP'd or patented. So it's an unknown estimate of the diversity. That's another problem. We don't know how accurate or inaccurate these data are when it comes to what was growing on farmers' fields. Okay, we go to China now. And pedigree data. Well, it may seem strange, but it's a better situation in terms of understanding diversity than the US. At least for the moment, up till now, the inbred parents of many or most hybrids grown in China have been publicly disclosed. Now, whether that will continue in the future, I have no idea. So the pedigrees of most inbred lines are publicly known, but again, the depth of information can be an issue. It's not so much of an issue at present because they are still relatively close to a lot of pretty old US germplasm. And I would assume that somewhere in China, there are data showing the relative usage of hybrids over time. But I haven't been able to find much of that when I've been looking, at least. So the pedigree data are a bit more, well, they're a lot more reliable for China. But again, if you want to weight the data in terms of how much acreage, that needs a bit more background research. Whereas in the US, it's basically impossible to fathom out. Now, let's go to marker technology. I've mentioned, again, SNPs are the way to go. And there's been this evolution through these different technologies. There are a lot of published studies from China on SSR and SNP data on several hundred of inbred lines. And there's 5, 6, or 7 studies actually that also include structure analyses that allow you to say, well, these are the major germplasm families used in China. So we'll get to those data. So the marker data for China actually are very good, and they're current. You're not disallowed by license or legal language from getting the profiling done and publishing the data. So that's quite encouraging in terms of understanding the diversity. So here, for example, is a look at a load of inbred lines from China. And so here are the major, so there's temperate tropic and tropical subtropical. Now, a lot of China, huge amount of production in China is temperate, most of it, I would say. And what I've done here is put in, these are the major groups I've listed on the left. And you're going to look at and here are some names that you know and you can understand. Ohio 43, A634, B37, B73, stiff stalk, non-stiff stalk, Missouri 17. This is being used in China. Okay. And on the left, I put in green the Chinese derived materials from Chinese land races. Remember, corn got to China very early in the 1500s. So they developed land races over several centuries. So there are two major Chinese groups here in green, and you see, as we mentioned, a whole load of yellow materials deriving from the United States. So there's a PA group and there's a PB group. And guess what the P stands for? Pioneer. There's a lot of stiff stalk from Iowa State. There's a heck of a lot of Ohio 43 and Missouri 17 in these backgrounds. So there's a lot known on genetic backgrounds of Chinese germplasm using SNP data. A lot more than that's publicly known for the US. And if you then look at the SNP data, and I use this publication Wu et al. 2015 because it had good coverage of time and it had a good coverage of widely used hybrids. Their structure analysis showed 7 major groups. And what I've done here is look at the most widely used hybrids in each of these periods and charted the change in usage dependence upon these backgrounds. So you can see that this TS2 is a Xingping Tao Chinese landrace up the top here. Less and less of it. HZS is another Chinese landrace. Fair bit of it, more, and then it's fairly consistent. But the thing is, from then you get down to the light blue, which is PA, which came from a Pioneer hybrid. And that was not much used up until the early 70s, and then has been much more use of it. Missouri 17, you might think this is really strange, and it is. I mean, this is a very old inbred line from what, the 60s, 70s, 60s. And look at the amount of dependence on it, even right through till now, or the 2000s. It's huge. Ohio 43 in gray, a slight decrease, but still quite a lot of use. I mean, this is going back to the 50s, I think, just like me, actually. I mean, good Lord. And then 207, well, 207 is also known as 165. It's a classic key iodent inbred line developed by Pioneer. A lot of dependence in the 60s, 70s, 80s, slight decrease here. B73, a bit, it decreased, and it seems to be coming back again. So these are the trends we see for China. Now, I mentioned about knowing exactly the area that hybrids are grown on. Now, there's 2 hybrids fairly recently developed, the Zhengdan 958, which is developed by a Chinese breeding company, and XY335, which is developed by Pioneer. And those 2 hybrids and descendants from those 2 hybrids are actually estimated to be grown over 85% of Chinese maize area. So that means that the dependence upon some of this HZS and all of this, particularly 207 perhaps, is really a lot bigger than it might appear if you treated every hybrid equally. Okay. So we've got some idea of the United States. We've got a better idea of China, and China is pretty similar to the United States and a little bit behind, because it's still quite dependent upon several old, in the backgrounds at least, but things are probably changing very fast now. Now, when you want to try and figure out the dependence or usage of different types of germplasm in other countries, it's difficult to impossible to find anything much. So you just surf the literature, and what you find is that Brazil, mostly tropical because it's a tropical environment, but there's quite a use of corn belt dent and stiff stalks introduced in the 1990s. Argentina, there's a lot more use of corn belt dents there, particularly since the 1960s. There's temperate growing environments, and so when you have temperate growing environments, you have a lot of corn belt dent. Europe, they had local flints. Corn got to Europe in the 1500s. They developed land races, but large-scale corn production in Europe only took off after World War II, and initially with a load of flints coming from Wisconsin, and then more corn belt dents, and particularly iodent, Pioneers 207 in the 1980s. Flints more to the north, corn belt dents more to the south. There are hybrids grown in northern Italy that would grow quite nicely in Iowa, for example. So a fair bit of corn belt dent in Europe. South Africa started using a lot more corn belt dents from the 1950s, and now there's a lot of corn belt dent in South Africa. So this just is this is an incredible summary, well, not incredible, but it's a very little summary of what you can find. That's from Pedigree data, and then this is from other data, including molecular marker data, but there's very precious little of that for these other countries when it comes to inbred hybrids used on farms. So when I say estimating, I mean sort of guesstimating, to be honest. And someone might criticize me for that, but that's the point. That is the point, that the information is not there. So we've already mentioned maize was expanded to the Americas, 350 races, 2% are economically important, corn belt dents most important. And looking at the United States and China, where there's the most information, that bears that out. Now, the global usage of corn belt germplasm is nothing new. I mean, that has been well established by many authors, some of who are listed here. Corn belt germplasm is well adapted to temperate environments. It is used in the subtropics. Dave Timothy looked into that. And it's being used by CIMMYT for improving yields in semi-tropical and tropical environments. So corn belt dent is widely used in temperate environments and increasingly in tropical and subtropical. Castro 2016 noted the use of temperate maize germplasm in tropical breeding programs has been poorly documented, but in general has been growing. Currently, many tropical hybrids contain some temperate germplasm. Looking at Eastern Europe and Russia, among other, looking at India, among other introductions into India, stiff stalk and Lancaster were used directly as commercial cultivars. And for one international commercial breeding organization, the similarity of hybrids to US temperate germplasm was estimated to have increased by 10 to 12% during the past 4 decades for hybrids developed in Brazil, Mexico, and India. So what I was trying to do here was just seeing if it's possible to sort of get a more recent estimate update of how widely used corn belt germplasm is, because getting back to this publication from 1972, they were concerned about the narrowing germplasm base of maize and other crops in the United States. So I think it's worth looking at the world, in fact. So this is what we're trying to do here. So this is the best we could do. So here we've got the top 20 producer countries listed. their production, the cumulative production, percent global production, cumulative percent global production, and then CBD, Corn Belt dent usage, percent country, then cumulative. And the little picture of me is obscuring the top right of my screen, so bear with me while I get my hard copy out here. Okay, so our estimate at the present time was that basically the United States is 100% Corn Belt dent germplasm, and percent global, that's 26.7%. China, we estimated at 80%. That is 15.1. So the cumulative then is 41%. And then it drops off, but it just keeps going up. And Brazil, 30% tropical, it's not so much. That adds 2% onto the global. Argentina, we put at 80% CBD, which adds 2.6% to global. So United States and China that produce 45% of production between them, most of that production is based on Corn Belt dent germplasm. And when you look at the top 20 countries, our estimate is that over half that germplasm is directly Corn Belt dent. And it's likely to increase. So there seems to be evidence that genetic diversity in the US is decreasing. Is this a so here's some questions. Is this a concern? If it is, what's to be done about it? What has been done about it? How effective has that been? Are there some new approaches? You look at global dependence on Corn Belt dent germplasm, and it's highly significant. Is this a threat or is it an opportunity? When you have international breeding programs, might it be that taking some temperate to a tropical environment helps the native tropical germplasm in those hybrids, and then you're effectively might be able to bring some of that improved exotic tropical back to the temperate environment, or does Corn Belt dent just wipe everything else out? I don't know. Is it or is it not important to monitor trends of diversity in breeding and cultivation? Then it begs the question: just what are the factors that contribute to agronomic performance? If diversity is going down in the US, yields genetic gain still seems to happen. How come? Why? But it costs more and more per unit genetic gain. Why? Is it just more difficult as you get nearer the top of a mountain, it gets more difficult to get further? I mean, when I started at Pioneer, the annual budget was $30 million. The entire budget of the company was about 35 million. Now, if you inflation adjust that, that's what, 50. Okay, well, you take today's budgets. Well, first of all, no one knows exactly what they are unless you were within the company, and then you might not. And then some of that, I don't know, half of it could be for chemical development, not for breeding. Then of the breeding part, how much is regulatory and GMO? But I would submit that still it's costing more per unit genetic gain now than it was in the 1980s. It's a good question as to why is that? And the question everyone looks at is, what does genetic diversity mean in terms of agronomic performance? And then it all comes down to genes times environment times management. And then where should people be investing? If you're the head of a large company, you first of all got to decide, well, where am I going to put my investments? How much goes into developing new chemicals? How much goes into developing new germplasm? If the people who make these decisions don't understand the potential that germplasm improvement can have, and I would submit most of them don't, then that sort of makes it problematic to get money flowing into the breeding side. And then when it does flow into the breeding side, how much goes into known genes, how much goes into anonymous genes, how much goes into trying to unanonymize anonymized genes, how much goes into GMO, to gene editing. I mean, these are really tough questions that all of the students at Iowa State and everywhere else who are going to be in the commercial sector will need to think about and have some good responses to. And then that, of course, relates back to, well, what's the research the university is going to do, and how does it, what education does it need to provide? I mean, these are really fundamental questions. So now we'll look at some of these things. So what are the factors that have contributed to US maize yield gains? Well, I think most of you all know that Don Duvik started doing genetic gain studies in Pioneer in the 1970s, and they are continuing, and others have done genetic gain studies too. His basic findings were that it's increase in planting density is a major factor. Now, is there an increase in harvest index now? We don't know. That's a good question. About, on average, 5% of total yield has been contributed by resistance to corn borer. This is protecting yield that, one way of looking at it is that this protects yield that was already bred into the plant. Some have, well, the analyses that was done in a paper I published, I didn't really understand the analysis, but I wrote it up anyway, actually prescribed about 1.3 kilograms per hectare per year was due to weather. And actually this publication by Butler came up with exactly the same number, which is quite amazing. But then we have a paper by Tolonar in 2017 that says 27% of yield trend between 84 and 2013 is due to solar brightening. Well, that seems quite a lot to me. Then we have a paper by Lobel and Burney in 2021 that says, well, air quality has improved since 1999, and that's increased maize yields by 20%. That seems a lot to me as well. So initially, it sounded somewhat of a relatively easy issue to measure genetic gain and ascribe what was contributing to it by planting hybrids developed in different time periods adjacent to each other in the same environments. But these more recent publications make that look a little bit more complicated. So I think we could do well with some more genetic gain studies. And it really, the US maize yields depend a lot on the environment. Ken Kassman, I guess, gave a seminar at Iowa State not too long ago, which people still talk about, where he said that climate and agronomy, not genetics, underpin recent maize yields. Well, okay, yes, in favorable environments. So when there's no stress, like as we found in Nebraska irrigated, the genetic gain is somewhat flat across years. But that is not the situation in most of the growing, most corn in the world is grown in rain-fed. And so to say that climate and agronomy, not genetics, underpin maize yields in favorable environments is all very well, but it doesn't reflect most of the reality. We know that marker-assisted selection, genome-wide selection has increased the efficiency and rate of yield of making genetic gain. But we also know from this study by Gaynor that unless you plug in some new diversity, you're going to lose genetic diversity. So again, that gets to, okay, you can more effectively use what you've got, but you better then start bringing some diversity in. Now, where that diversity comes from and how you do it is another question. So that's that's a critical issue to think about. Now, there are several programs have been initiated, particularly since this report in 1972 that I mentioned, GEM. If you don't know about GEM and you're an Iowa State student, shame on you. You better learn about it real fast and go to the PI station. North Carolina State University, Major Goodman has a program adapting tropical inbred lines to temperate environment. He participates with the GEM project. North Dakota has a program. They've demonstrated there is positive potential, but this is the trick. It's not positive enough to actually fuel an immediate release into commercial products. So, but unless you have a longer term program to bring in new diversity, you won't have a short term. But you've got to have a short term, else you can't have a long term. I mean, so this is what you've got to fathom out and get the balance right when you're managing a breeding program. Now, what has been learned from these programs? We've got an NAPB meeting coming up soon at Iowa State, and part of what we can talk about is, well, what's been done and what have we learned? As I mentioned, maize breeding in a tropical environment, if you have an international breeding program, is that pre-breeding for the temperate conditions. And might gene editing be used to rapidly adapt tropical germplasm so that it actually flowers and can be evaluated in temperate conditions? That may be really, really important. Looking at diversity in the US, this is data from Van Heerwaarden, where they looked at SNP distances between inbred lines from 3 different eras, the 50s, the 80s, and the 2000s. And there's basically, and they looked at stiff stalks, non-stiff stalks, and iodents, and there's basically a decline in diversity as it's measured this way. And if you just plot this out so simplistically as to be ridiculous, we've already run out of non-stiff stalk germplasm, and that 10 years ago. And soon we'll run out of stiff stalk germplasm. Well, then if you've just left with one germplasm pool, bye-bye hybrids. Now, this is grossly oversimplistic, but you can't ignore it, I don't think. Well, you can, but I think at one's peril. And this, in fact, is the last survey that was done of widely used maize hybrids that were on farms in 1991. And this is from a crop science publication. And goodness knows what the situation is now. I have no idea. Are we happy with it? We've no idea. I think we need to discuss that. And as I mentioned, the actual information known about pedigrees that's publicly disclosed is going down and down and down. So this is a figure from White et al. In Crop Science 2020. And you can see you've got 1980, 90, 2000, 2010. You've got different companies, and you've got these different germplasm backgrounds here. And in 1980, there wasn't much gray. I mean, there wasn't much, well, there was hardly any unknown. It was all, could all be accounted for by these different backgrounds. 1990, then there started to be some gaps, and they've only just increased. So the usefulness of pedigree information to chart genetic diversity is absolutely useless, basically. So you've got to use markers. And that's what I've got to present. I hope we've got time for questions and discussion. Thank you.
Okay, well, thank you for that seminar. Are there any questions? I'll start with one. In the slide that you showed usage of different germplasm pools from China, there was a B73 group.
Yeah.
That seemed to precede the release of B73. It showed a use of that group. So I'm wondering if maybe that's really a B73, that one, you just went by it.
Yeah, I've got it here. Well, it says B30. Oh, sorry, that's B37. There's B73. Oh, they want the next one.
Yes.
Okay.
The B73, which is labeled in blue, B70. So I'm wondering what that means in the 1960s.
Well, you've got me wondering now, so I better go and look at that. I'll need to check that out, Jody. Thank you.
The other thing I wondered a little bit about, it's the amount of Ohio 43 and Mo 17 here really are stunning.
Oh, it's amazing.
Because Mo 17 is rapidly gone the other direction in the US.
Absolutely, yeah.
And I'm just wondering, perhaps, the HZS group, that could have something to do with local adaptation. I mean, that's a big deal, but the PA group.
You would think. Yeah, but the Ohio 43 and Mo 17 is real. I mean, they were using those inbreds directly for 2 decades at least.
Wow. So
103 even.
Perhaps there was something about those inbreds did well in China. The other question I had, though, the PA group, that's a pioneer group. I mean, could that include also some contribution of stiff stalk or other groups, so that perhaps, it doesn't look like there's a lot of B73 in here and some of these others, but maybe with that PA group and the IDT, the PA group could include some things that are stiff stalk and or IDT, which would perhaps make those groups a little more important.
The PA and the PB groups actually come from hybrids. So immediately you've got two heterotic pools that funnel into PA and funnel into PB. And so one of the difficulties China's had is that it's had too many heterotic groups, and they're not well defined enough, and they haven't made the most effective use of them. Now they're beginning, I think, to narrow it down a bit now. But the PA would be a mix of stiff stalk and non-stiff stalk, and the PB the same. And by selection, they might have gone one way or another, or both. But that's a problem in Chinese germplasm when they're developing from hybrids. It keeps these heterotic groups being less systematically useful and not so well characterized as they need to be. That they've got to. That is one area of improvement, I think, for hybrid breeding in China, is to get the heterotic groups sorted out a lot better.